in bats in Mahabaleshwar cave antibodies against Nipah virus. Till date, India has experienced 4 episodes of NIV outbreaks with CFR ranging from 65% to 100%. The first evidence of NIV infection was reported in Siliguri district, West Bengal in 2001, followed by Nadia district in West Bengal in 2007. A cross-sectional survey by Indian Council of Medical Research, National Institute of Virology to study the prevalence of Nipah virus (NIV) in bats of India has picked up samples with the presence of antibodies against Nipah virus in some bat species from a cave in Mahabaleshwar, a popular hill station in Satara district, Maharashtra. NIV is one of the top 10 priority list pathogens identified by the World Health Organization. Till date, India has experienced 4 episodes of NIV outbreaks with CFR ranging from 65% to 100%. The first evidence of NIV infection was reported in Siliguri district, West Bengal in 2001, followed by Nadia district in West Bengal 2007. The presence of NIV antibodies were detected in Manuguri and Dhubri district of Assam and Kuch Bihar of West Bengal, both places situated close to Bangladesh border. A third outbreak occurred in Kochikode district of Kerala state in 2018 with 18 cases fatalities, followed by another outbreak in the same state in 2019. A study in 2018 has identified many Southeast Asian countries including Indian states as potential hotspots for the NIV disease. Petros medius bats, which are large fruit-eating bats, are the incriminated reservoir for NIV in India, as both for NIV RNA and antibodies were detected in the samples of this bat collected during previous NIV outbreaks. Studies on other species of bats as potential NIV reservoirs in India are very limited. The new study detection of possible Nipah virus infection in Rosetus lecentioli and Pteris pipistrellus bats in Maharashtra, India, published in the Journal of Infection and Public Health, has found the virus and antibodies in different species. During March 2020, from a cave in Mahabaleshwar, 2 species of bats, Rosetus lecentioli, medium-sized fruit-eating bats, and Pipistrellus, tiny insectivorous bats, were trapped by researchers using mist nets. Blood, throat, and rectal swab samples were collected on site from anesthetized bats. Throat and rectal swab specimens were collected from all the bats. Necropsy of 10 bats of each species was performed at the containment facility of Indian Council of Medical Research, National Institute of Virology, Pune. RNA was extracted from samples and anti-NIV-Ig antibodies were detected in a number of samples. 1 bat each from R. lecentioli and PP pestis species tested positive for both NIV RNA and anti-NIV Ig antibodies, the study said. This is the first report of possible NIV infection in R. lecentioli bats in India, which was demonstrated by the presence of both NIV RNA and anti-NIV LGG antibodies in bats, said Dr. Pragya Yadav, NIV scientist and one of the authors. The cross-sectional survey was initiated to study the prevalence of NIV in bats of India by random sampling of P. medicis, R. lecentioli, and P. pestilens bats that have widespread prevalence in India, said Mangesh Khokhle, lead author of the study, and Dr. T. Moria, former director of NIV and one of the authors. In earlier investigation during the last decade, NIV activity could not be detected in R. lecentioli despite processing several hundred bats, including bats from the same location. The exposure of R. lecentioli bats to NIV warrants further investigation as roosting and breeding habitats of the refugees and very rarely, the scientists said. More studies in bats and humans are therefore needed to understand the prevalence of the virus in the state. The roost was sampled was age-old and the virus might have been circulating among the inhabitants at low levels and not detected during early studies. Alternatively, a new introduction might have occurred from P. medicis to rosetus bat through NIV contaminated fruits, as both share the same fruit trees. NIV detection in P. pisciculus bats and insectivorous species and their role in virus spillover to human appears remote, the study said. Their positivity might be explained through sharing the same habitat with R. licentioli bats inside the cave. Researchers said it was difficult to infer any conclusion as only a few bats were screened during the present study. Recurring outbreaks, high case fatality rate, human to human transmission, and lack of effective vaccination antivirals pose a major concern in India, as bat roosts are very common in areas where large human population reside. It said. Thank you.